right, so today we're doing transport across the membrane. Last time we talked all about the membrane. Oh, I have to turn this on for. Last time we talked all about the membrane. What is the membrane made of? What three components? Carbohydrates, lipids, and proteins, and specifically what kind of lipid? Phospholipids. Now, phospholipids are super cool and important because they form these bilayers. Why do they form bilayers? They're amphipathic, which means they are both attracted to and repelled by water at the same time. So the polar heads orient toward the water, the nonpolar tails orient away from it, forming these double layers. All of your biological membranes are going to be composed of these phospholipids, whether they're the outside plasma membrane of the cell or the membranes that make up the organelles. The phospholipids, where are the phospholipids built in the cell? Phospholipids are built somewhere in the smooth endoplasmic reticulum. Now, these membranes, this plasma membrane that surrounds the cell has a whole bunch of functions. Give me some of the functions of the plasma membrane. Cellular adhesion, cell signaling, movement, structure, but probably the most important one it has is selective uptake. It will take up some things but not others, or it will release some things but not others. The proteins inside that are embedded within that membrane help to give it some of those selectively permeable properties. Today, the whole lecture is all about getting transported across the membrane. So we're going to talk about the process of diffusion, osmosis, and active transport. So first off, we got this term, I've been throwing it around fairly loosely. It's called selective permeability. You let some things in, but you don't let others in. The cell membrane is capable of regulating what molecules come in and out of it. It prevents other molecules from doing so. So it is selective. Think about, and a lot of times I think about this as you let people into your life. Now, you want to regulate who you let into your life. You meet a whole bunch of people during the course of your day. Random people at the store who tick you off, that person who tried to steal your parking spot, bosses, whatever. All of these people are people who you have to deal with, but you don't want to really get to know them. You don't want to let them into your inner circle. You're selective about it. You're going to date somebody. You're not going to date whoever comes along. Or maybe you do. I don't know. You do your thing. You have to allow some things in but not others. And the cell has to do the same. You don't want it to allow in these chemicals that might hurt the cell. You only want to bring in those things that can help. There are three big ways across the membrane: diffusion, facilitated diffusion, and active transport. We have to get across the cell. What makes it so very difficult for molecules to get across that membrane is that hydrophobic center. You've got these phospholipids with the hydrophilic head, hydrophobic tails. The molecules that are already dissolved in water are very likely to be either hydrophobic or hydrophilic. Which one? If they're already dissolved in water. Hydrophilic. If they're hydrophilic, they're dissolved in water, they can very, very easily interact with the polar heads of the phospholipids. Not so the polar tails. So because of that hydrophobic region, these molecules have a hard time getting across the membrane. In this case, I'm looking at Tootsie Pops. You guys know the Tootsie Pop thing? How many licks does it take to get to the center of a Tootsie Pop? Do you guys ever test that? I'm sure at some point in your life, you have sat there with a Tootsie Pop and you're like, I'm going to figure out how many licks does it take to get to the center of a Tootsie Pop. You're telling me none of you have done that. Thank you. We're going to get some Tootsie Pops. We're going to do that. And then you'll have some sticks to play with at the end. It'll be good. Works out. Win-win for Janie. So it's the idea that that center is very different than the exterior. The exterior has one set of properties. For a Tootsie Pop, the exterior is the hard candy shell. The interior is what's ostensibly a Tootsie Roll. Probably not. They taste totally different. But still, one part of it's different than the other. What interacts with one easily does not interact with the other. Some molecules can go straight across the membrane. These are what are termed highly permeable. Gases are highly permeable. Think oxygen, carbon dioxide. These small molecules are transported across the membrane as if the membrane didn't exist. They fit in between the phospholipids, and it's almost like there's no barrier at all. Small, very, very small uncharged molecules do the same thing. If they're uncharged, they're not attracted to the polar heads. They're able to slide right in between the phospholipids. This comes into play very heavily when you talk about the human body. Do you guys know when you take a breath? All right, so you guys have done this before. Do you guys know basic human biology? Some of you guys do. You take a deep breath. What goes into your lungs? Good enough. Oxygen, big mix, but oxygen's the big thing. Oxygen's going in there. It's hitting your lungs. Your lungs have these branches. Those branches have little tiny branches, and those tiny branches have these little like grape sacs on them. Blood, so cells are going all around those, constantly moving. And as they're moving, the oxygen is moving from the highly oxygenated lung to the less oxygenated cell, across the barrier as if it didn't even exist. At the same time, your red blood cells are bringing carbon dioxide up. That's going from the high area concentration of carbon dioxide in the red blood cell over to the low area of carbon dioxide, low region of carbon dioxide in the lung. So you get this gas exchange. That's really important for later in this sequence. Some molecules are moderately permeable. They're able to get through, but not as easily. One of those is water. Now, it's kind of surprising that water's at all permeable. Why is it surprising that water can get through this thing at all? I mean, I'm telling you, water can get through, and it can get through almost as if there was no barrier. How is it that water molecules can, why is it weird that water molecules can get through so easily? Right, you got this big hydrophobic center that should act as a shield bouncing these water molecules off, but it doesn't. Think about it this way. Water molecules, if this cell's sitting in an aqueous solution, are absolutely, completely, and totally surrounding this cell. The whole cell's absolutely surrounded by water. There are billions upon billions upon billions of water molecules. If even a small percentage make it through, that's still a lot. A small percentage of billions and billions and billions and billions is still millions and millions and millions and millions. It's like water's flooding through constantly. So you can think about it this way. Most of the time, water's coming in, hitting the hydrophobic layer and bouncing off. It's almost like, do you guys remember those when you play with Hot Wheels and there's these little spinny things on the side of the track and you put the car in and it shoots through? You know what I'm talking about? Okay, good. Whew. At least you're not laughing at, like, yeah. So, yeah, spinny thing. You put the car in, it shoots right through. What if you have the car going in the opposite direction? You push it at the spinny things. Well, it's not going to go rocketing through if the spinny things are moving in the opposite direction. That's right, it's going to bounce off for the most part. You hit it, it hits it, strikes it, and it flies off in some sort of weird direction. And you guys know, if you ever played with one of those, that was the second thing you did. The first, of course, was you turned it into some sort of, I don't know, car shooting gun and you chased your siblings around the house. But for this particular instance, think water molecules are coming in, they hit that barrier, and they're deflected off immediately. Sometimes, though, water gets in, and instead of being deflected off, it gets accelerated forward. So it's going to hit, almost like hitting those spinny things going in the right direction, and gets rocketed inside. So water has this, it shouldn't get through. But because some of those molecules get through, some of this a small fraction of this large number of molecules get through, it's as if this is permeable to water, letting water go backwards and forwards pretty easily. So water has moderate permeability. As far as low permeability, things that don't get through often, you have polar organic molecules and sugars. You guys know sugars. What shape are sugars usually? Two types of shapes. Hexose and pentose. They're round. They're big. They don't fit between the phospholipids. Oftentimes they're charged. If they're charged, if they're polar, they're going to interact with the hydrophilic head, but they'll be attracted to it. So they're not able to get through. They've sort of stopped at the border. Then you've got these extremely low permeability molecules. Charged polar molecules, amino acids, proteins, nucleic acids, polysaccharides. A lot of these are just gigantic. You can't get them into the cell. They're just not able to get across the membrane. They're too big. They don't fit. So as far as our we have high and moderately permeable. You have gases will go right through the cell. Water, a little bit of slowdown, but it can still make it through. Small sugars, things like that, they can make it, but it's getting really hard to get through the cell. And things that can never get through the cell are going to be like nucleic acids, charged things, proteins, amino acids. They're too big. There's a different way through for them. All right, so we can get through the cell. Let's talk a bit about diffusion. How do these things move around? Where is a piece of chalk? Yeah, but that's so small. Here's a bigger piece. And here's an entire chalkboard for me, too. And a bunch more chalk, apparently. All right, so you've got these molecules. Individual molecules. Doesn't matter what they are. Solutes. You have solutes and solvents. The solvents, what the solutes are dissolved in to create a solution. In this case, we have on one side a whole bunch of solutes. Lots and lots and lots of them. On the other side, not very many. This forms what's known as a gradient. You're going to have a high concentration on one side, a low concentration on the other. That's it. If there's a membrane in between, it's called a transmembrane gradient. Now we could draw out these little circles, or we could just label them. We could say that high concentration is, say, 1 molar. Because remember, a mole is just a measure of the number of solutes present, the number of molecules. So 1 molar, 0 molar. And what you find is that the solutes tend to migrate toward where there's a lower concentration. That process is known as diffusion. So if you have a high concentration on the left side, a low concentration on the right side, the molecules will move all on their own to the right side. Questions on that? Okay, so reminder, in case you forgot that water lecture because it happened before the last exam, you have a solute, which is the thing that you dissolve, the solvent, the thing that does the dissolving, and they come together to form a solution. What do you call solutions that are made using water? Aqueous solutions. So in this case, we've got water as the solvent, hot chocolate mix is the solute, and the solution is hot chocolate. When you have a gradient form using ions, so on one side you have a lot of positive ions, the other side you have a lot of negative ions. You have formed what's called an electrochemical gradient. This is the first introduction to electrochemical gradients, but they're going to be critical for forming energetic reactions in the body. Without electrochemical gradients, you would not really be able to make energy. So you can form gradients not just based on the number of molecules present, but the charge of those molecules. And if you form it based on the charge, it's known as an electrochemical gradient. So in this case, you've got the sodium, so Na plus wants to move from left to right, and the chlorine wants to move from right to left. That process of moving across a gradient, dispersing out, is going to be called diffusion. Now, if you stick a membrane between them, these membranes are selectively permeable. They may not allow the solutes through, but they will allow water through. So even though the solute wants to move from one side to the other, it can't. Instead, the water molecules will move. So that membrane stops solutes from moving across, but will allow water. Now that we have a barrier in the way, we can start describing our solutions. A solution will be termed isotonic. Iso means same. Think isosceles triangle, for those of you who are good at math. When the solute's concentration on the outside of the cell is the same as on the inside of the cell. So you have isotonic solution is the same solute concentration as the cell. A hypotonic solution. Hypo means small, little. A hypotonic solution has less solute than the solution it's compared to. So in this case, the cell, this orange piece, is placed into a hypotonic solution. The final category is hypertonic. With a hypertonic solution, the cell is placed into, the orange one, is placed into a solution, the blue, that has more solutes than it. Greater solute concentration. So let me ask you, here is our cell, here's water. Inside the cell, you have a 2 molar solution. Outside the cell, you have a 1 molar solution. What would you label that solution on the outside of the cell? How would you describe it? Hypotonic. It's got lower solute concentration. Questions? All right, so we're talking about the difference between hypotonic, hypertonic, and isotonic. The cell here, this round part, has 2 molar solution. That means it has 2x number of molecules. The solution is a 1 molar solution. How would you describe that solution? And you describe it as hypotonic, because there are less solutes outside than inside. Okay? Now, water molecules are going to follow a concentration gradient. They're going to move from an area of high concentration water molecules to an area of low concentration water molecules. That's the official definition of osmosis. You could also look at it in the way I tend to prefer to look at it, as moving water from a hypotonic solution to a hypertonic solution. So if you write nothing else down, write down that water moves from hypotonic to hypertonic. So what would happen to this cell? It's got 1 molar solution, 2 molar inside the cell. Where's water going to move? I am not trying to trick you. That's right. Water will always move from a hypotonic solution to a hypertonic solution. What's going to start happening to that cell, do you think? It's going to start expanding. So you're moving water across that membrane, and that's in an attempt to balance out solute concentrations. I was very proud of this particular slide. It's got animations and everything. I never have that. But as you can see, on the left side, you've got low solutes. On the right side, you've got high solutes. Water is moving from the left side across the semi-permeable membrane to the right side. It's semi-permeable because it's letting water through, but not the other substances. What would you term the left side? Hypotonic. And the right side then? Hypertonic. It will continue proceeding until they balance out, until both sides have the same solute concentration. At which point, what would you term it? Isotonic. Now this osmosis, this movement of water across the concentration gradient to reach equilibrium, requires absolutely no energy. It's termed passive. Passive transport does not require any energy whatsoever. So here's an example. Water is moving into the cell. What can you tell me about the solution? Which way does water always move? Hypotonic to hypertonic. So therefore, the solution outside is what? Hypotonic. So what's going to happen to the cell? It swells. Here, water's leaving the cell. What can you tell me about the cell? It's hypotonic compared to the solution, which is hypertonic. What's going to happen to the cell? It's going to shrink. Here, you have water moving in both directions equally at the same time. What do you call this? Isotonic. I guess a practical application of this is if you think about, have you guys ever seen a slug before? All right, you've seen slugs in all sorts of places. If you ever put a pumpkin out for Halloween, you'll notice that it's being eaten in little chunks by slugs. What do you do to a slug if you want to kill it? Put salt in it. Yeah, all you guys are like, I don't want to say I do it, but I totally do it. When you have the salt is a solute, right? You take salt, you put it on a slug. You've created a hypertonic solution around the slug. So what happens to all the water in the slug? That's right, it goes out. And what happens to the slug? Shrivels up. You're removing all that water using solutes. All right, let's go the other way. Have you ever accidentally, you found a slug, in your cat's water bowl or something? No, of course not. If you've ever found a slug in a bowl of water or in a bucket of water, what do they look like? They look big and fat and dead. Why do they look big, fat, and dead? All of that water flooded into their cells. Think about your fingers. You guys, you do this. You've taken showers before, I assume. When you take showers, have you ever taken that super long shower that felt so good you didn't want to get out? And you come out and your hand's all wrinkly, right? So you've been sitting in this pure water, no solutes. So what's happening to your cells? What's that water doing to your cells? Pure water, hypotonic solution. Your cells, hypertonic solution. Which way is the water moving? Inside the cells. So your fingers start expanding. They're pushing against the collagen and giving you this little wrinkly look. And then when your cells dry, when they start losing that water, it goes back to normal. Water moves from hypotonic solution, pure water, to a hypertonic solution. In animal cells, you've got crenation, where a cell will crenulate. And I use the word crenulate and crenation. So crenation, Dr. Balfour and I just had a discussion about the proper use of the terms crenation and crenulation. The process is crenation, and a cell is crenulated. But that means that if you stick a red blood cell, say, an animal cell, into salt water, all of this water will start to leave the cell, making it shrivel up like a little raisin. Not good for you. You guys know not to drink salt water, right? Pretty bad for you. Don't do that. You drink a cup of it, you die. On the other side, you take one of your cells and you put it into pure water, no solutes in it. Suddenly you've dropped your red blood cells into a hypotonic solution. Water will flood into them and you get what's called osmotic lysing. That is, the cells burst. You don't want that to happen. So in animal cells, if you were to put it into a hypertonic solution, you get crenation. If you put it into a hypotonic solution, you get osmotic lysing. Animal cells need just the right conditions to survive, although they do have some defense mechanisms for this. Plant cells, on the other hand, if you were to add water to a plant cell, pure water to a plant, what happens? What do you water plants with? You take pure water. Don't use Gatorade or Brondo. Pure water. You add it to a plant. What happens to the plant? It grows. It's all happy. It's got this it's what's called turgid. It's rigid. Plants looks very healthy. It's sticking straight up because it's got this cool cell wall surrounding it. That cell wall prevents it from popping. It prevents osmotic lysing. The cell wall, when the cell gets filled with water, it pushes against the cell wall, making it very, very rigid. The other side, what if you were to water a plant with salt water? It wilts. It wilts very, very quickly. The water is removed from the plant cells, and you get what's called plasmolysis. The plasma membrane rips off of the cell wall and shrivels up in a corner. And I believe you guys looked at that last week in lab. So you could have turgor if there's a lot of water present, or plasmolysis if you have a lot of solute. You'll notice that with nurses, if you're dehydrated, they're never going to hook up a big bag of sterile water to your arm. People have thought that. They're like, well, I need water, so just put it right in my vein. It's a bad idea because then people die. The reason it's such a bad idea is pure water does not have any solutes. Your blood cells do have solutes. So what will happen to your red blood cells if you were to surround them in pure water? Osmotic lysing. They will all start popping. And then you have a lot of paperwork on your hands. Instead, you hook up what's called a saline solution. The saline solution is specially mixed to have the exact same level of solute as your blood cells. So you're replacing fluids while at the same time not allowing your cells to pop. Kind of important thing to know if you're ever going to need to resuscitate somebody. Now, obviously, there are some animals that survive in water. They can do this, especially you guys saw a whole bunch of protists last week that were able to survive in water. How do they protect themselves? How can these protists stop themselves from popping? Contractile vacuole. A contractile vacuole is going to fill up with water, and then it's going to use energy to push that water out. So water is constantly flooding into these protists, filling up the vacuole with water. Before the cell can blow up, though, the animal is able to use that use energy to squeeze the vacuole and push the water out of it. It's almost like a sump pump for your house. So there we go. That's where we've got osmosis, the movement of water molecules across the membrane. And we alluded to it earlier. The flip side of that is the movement of solutes. As you're moving a solute, that process is known as diffusion. We're going to move solutes from an area of high concentration to an area of low concentration. We're no longer talking about hypotonic, hypertonic solutions. We're simply talking about the level of solutes present in any particular region. So at the top, you'll notice you've got a whole bunch of dye. That dye is orange. It's on the left side. Given time, those orange molecules will spread out until there's equal distance between each. If you look at the bottom one, here you've got a lot of dye purple dye on the left, orange dye on the right. Given time, they're going to spread out to where there's an even number of purple ones on both sides and orange ones on both sides. You guys ever made Kool-Aid? Not, this works for tea as well. If you think about Kool-Aid, you take Kool-Aid, you can take, it's mainly sugar and dye, right? You take a thing of Kool-Aid, you dump it into water. Where does all of that dye fall? To the bottom. It sits at the bottom until you stir it up. But let's say you're lazy and you don't ever stir it up. Given time, the lazy man can just prep their Kool-Aid, set it aside, and walk away. 2 days later, they come back and it will be entirely mixed just due to diffusion, just due to the spacing out of these molecules. You guys ever made, I guess they call it sun tea? It's okay. You ever made sweet tea? Okay, a lot of you guys have. It's so weird. I go around here and I order a tea, and they always bring me sweet tea. And I'm like, no, I want tea, not this. Yeah, I know. You have to say unsweet tea around here. You don't have to do that anywhere else in the country. So the way you make it is you take these bags of tea, right? Or even just leaves of tea, and you put them into a large container. Over time, starting first where those tea bags are hanging out, there gets to be a brownish cloud. That cloud expands. Now, it's a really dark brown at first, and then it expands, and then suddenly everything is the same color over a long period of time. That's due to diffusion. If you heat it up, it diffuses even faster, because now you've excited water molecules. They're moving much more quickly than they were before. Cool it down, they move more slowly, you get slower diffusion. That's why people will put tea in the sun in order to get it, iced tea in the sun in order to get it to mix better. So here's some examples. Let's just define some colors real quick. What color is this? Good. What color is this? And this? Okay, thanks. I just want to get on the same page with those. So we've got blue, red, and green, because some classes, they get really upset when I accidentally call that purple. I don't know. Well, now you guys are like, well, now we're fighting over this. But it's kind of purple. But, all right, so let's say we have these blue or purple things. Which way are they going to move? They're going to head over to B. How about the pink things, pink or red things? They're going to head toward A until they're evenly spaced. What about the green things? Yeah, they're pretty diffused already. So wherever you have a high concentration, it's going to move to that lower area of concentration until everything is equally spaced, until you're in a state of equilibrium. So that's osmosis and diffusion. Both osmosis and diffusion are passive transport. And again, what does it mean to be passive? There's no energy required. Now we're going to start talking about some of those molecules that can't just make it across the membrane, that have to rely on proteins to get across. You have different types of proteins that help you get across the membrane. They alter the permeability. These proteins may allow some molecules through that the membrane itself would not. So you have channel proteins and transporter proteins. These proteins are embedded within the plasma membrane. So what would you term them? If they're actually embedded in, if they are part of the membrane. Starts with an I. Integral membrane proteins. If they go all the way across, what are these integral membrane proteins? Across means trans, so transmembrane. So first up, we got our channel proteins. Channel proteins form a channel, like the English Channel. Think of a waterway. They're transmembrane proteins. They go all the way through the membrane, and they have an open structure. So anything that's on one side can flow straight through to the other. It's like a water slide or a tube in Mario Brothers. You go from one side to the other. That means that if there's a lot of solute on one side, so hypotonic solution on the, hypertonic solution on top, hypotonic solution on the bottom, The solutes will want to move from the top to the bottom. The water wants to move out. They are not. There are certain channel proteins that are selective, but a good number are not necessarily selective. For a selective protein, you're going to look at your transport proteins. For channel, think just big open things that will allow whatever through that can get through. One thing that will always be selective on these, though, is the size. So some of the channels are much smaller than other channels. Next up, you have your transporter proteins, like your carrier proteins. I was looking at these like gated communities. Don't ever write gated communities on your exam. That's not a type of protein. I don't know. Half of my class wrote that one year. By gated communities, what I mean is there's a stop. There's something to prevent other molecules from getting in. It's not an open channel. It's much more selective. So you have these carrier proteins. One type of these carrier proteins is called the uniporter. Uni means what? One. In this case, you're going to transport one protein across the barrier. Here, this piece of chalk is my protein. It's on one side of the barrier. As the carrier protein, my job is to grab it, bring it across, and drop it on. Oh, man, I threw the chalk. Drop it on the other side. Got it. One molecule binds, gets brought over, and then released. In that process, there's no big opening between the two sides. It's still closed the whole way. It's almost like an airlock. It comes in, it binds, it turns to the other side, it's released. A SYMPORTER. Sym means together. In this case, you've got two molecules that are going to bind to one protein complex. So you have one protein comes along and binds. So I got two here. One protein binds. That opens up a second receptor site. The second protein binds. Once these two have bound, they can then go and be let into the cell at the same time. Both are required to go together. They can't go just one at a time. You will only ever have two. You will only have them coming in the same ratios. That maintains the proper amount of one molecule in comparison to another molecule within the cell. If I need to make, let's say we're making salt in our cells. We're not, but let's say we are. I would grab sodium, Na, and then I would grab chlorine, Cl, and I would bring them both through together so that they can form NaCl. I don't want to bring in 15 NAs and then one Cl. That's not how these SYMPORTERs work. They bring in molecules in a specific ratio to each other. And then the final category is the antiporter. Antiporters are going to let one molecule in for every one molecule it lets out. So you're bringing one molecule through. That means you're going to push another molecule out in the other direction. Again, you're maintaining ratios. So you have your uniporter, one uniporter carrier, which is going to carry one molecule across the membrane. Your SYMPORTER, which can carry two or more across in the same direction across the membrane. And the antiporter, which takes two or more molecules across in different directions. Now those are the only procedures, diffusion, osmosis, and some types of carrier proteins work through passive transport. They're using a concentration gradient. But those processes that move against a concentration gradient are going to require energy. They're termed active. So active transport, moving against a concentration gradient, requires energy. That's kind of a big concept there. Moving against a concentration gradient requires energy. So I put this this quote up here, "Thus we beat on boats against the current." Have you guys ever heard that quote before? I think I've had a grand total of 1 student who's ever read the book or who remembers the book that came from. Oh yeah, totally. The Great Gatsby. You got that. There was a point when I first started teaching here, I was disappointed that nobody knew that. And I was like, I will give an A to anybody who can get where that quote came from. And then I was like, good thing they didn't get it. But it comes from The Great Gatsby. Have you guys ever read that book? A lot of you guys read that book, I guess, in high school because you were forced to. It makes a lot more sense as you get older. Reading into it, you see things, you start to see, unfortunately, you see things in yourself. So if you haven't read The Great Gatsby, you guys have watched the movie, though, haven't you? Yeah, some of you guys are nodding. Sorry about that. The book is really good. The movie, not so great. The book should be mandatory watching because pretty much the book is all about, do you guys remember the book? The storyline? You got Gatsby. Gatsby sort of faked his way up to being at the top in order to get with the girl that was already married to some other guy. Yeah, that was dumb. What happened to the end? What happened to Gatsby in the end? Yeah, dead. So really what it comes down to is if the person you're interested in is with somebody else, just give up. It's okay. Move on with your life. Forget about the green light at the end of the dock. Primary active transport is going to use energy. A protein will use up energy in order to move molecules against the concentration gradient. If you were to go, hypothetically, if you guys were ever to do any sort of water sports on the Dan River, because you don't worry about cancer, you're going, let's say, rafting down the river, or canoeing down the river, kayaking down the river. I've seen people do that. That was dumb. They're kayaking down the river. If they go with the current, they don't need to use energy, right? That's totally passive. You could just sit there with a paddle in the boat and look around. But what if they go in the other direction? What do they have to do? They have to row. They got to paddle like crazy. Sometimes that current gets strong. They need a motor sometimes. So we've got active transport is going to need energy in order to move against the flow of the concentration gradient. So here we've got the blue ones. You've got lots and lots of blue ones at the top, few blue ones at the bottom. Diffusion says they're going to go straight through. No energy, passive. On the other side, we've got very few red ones, lots of red ones at the top. The only way to get the red one up that gradient is to pump it out, to use energy. We have some transporters that are super awesome. They use a pre-existing solute gradient to power a pump. So that means that you're not going to need to break up and use cellular energy. You're using the gradient that's already around you. Easiest way I can have you guys picture this, well, one is this prettier slide. You've got the red ones. They have a high concentration at the top. They're pushing down on this molecule that will spin. The yellow ones then can be ejected. And that's like an antiporter. One comes in, one gets pushed out. You use the energy of a gradient for one to push the other out. The other way I think about it is a revolving door. You guys played with revolving doors before? Gone to a hotel and you're just like, I'm going in a circle. And you got that very narrow window at which to get into the revolving door. Perhaps as you've done this, you've realized you don't need to push on the revolving door as long as somebody else is coming in the other direction. Have you done that before? Thank you for admitting that. I'm totally a mooch. So I've got my, my bags as I'm leaving a hotel. I go into the revolving door, and I'll just wait. People look at me like it's crazy. But I was like, somebody will come along in a second. Somebody comes the other direction, and they start pushing on the door. Now I don't need to push on the door, and I can go right through. It required no energy on my part. The other person, now they had to use energy. That's like secondary active transport. You can get one molecule through using the existing concentration gradient going in the other direction. This is going to be super important when we start talking about cellular respiration, because you can use secondary active transport. You've built, here's the sneak preview, you're going to build an electrochemical gradient using ions on one side of the membrane. You're going to then use secondary active transport to move those ions across the membrane. And then as that protein spins, it produces energy, almost like a a water wheel. And we use that energy to create energy intermediaries. And all of that happens in the mitochondria. I just couldn't wait to tell you. Don't worry, we'll do 2 lectures on it. So by compounding these secondary active transports with an electrochemical gradient, suddenly we can power all sorts of biological reactions. Suddenly, you have energy to do things. You've taken sugar, this crystalline substance, and broken it down to form usable energy. So there we go. You can get through, you can change the permeability using proteins. You've got water that can make it through the membrane, and then you can diffuse some solutes across it. But then we have those molecules that are just too big to get across. DNA, polypeptides, amino acids, nucleic acids. They're too big, they're too charged, and they're not going to make it through. So we need to get them into the cell without going through these channels. One way we do that is through a process known as exocytosis. We've talked a little about this before. This is where material from the inside of the cell is packaged and then shipped to the outside of the cell. Where is this going to be packaged? Where is the final sorting station of the cell? The Golgi apparatus. So you've got your Golgi apparatus forming these membrane-bound compartments. What are these compartments called? They're filled with stuff. Vesicles. And they're going to move from the Golgi toward the membrane. All right, what are they riding along? Microtubules. And what's carrying them? Motor proteins. They get over to the membrane. And when they get to the membrane, they fuse with the membrane. And they release whatever they had stored. Proteins, amino acids, nucleic acids, whatever. Anything big. So sort of like, here's my polypeptide. I encase it in a vesicle. I move it from the Golgi apparatus over to the membrane. The vesicle fuses with the membrane and begins to open up, and then it pushes those molecules out of the cell. Exo, think out, exit. Cytosis, the cytosol. So you're leaving the cytosol. The flip side of that's called endocytosis. Endo means in. So here we're taking particles from the outside and bringing them in. You'll notice in phase 1 you've got a food particle in the cell. Phase 2, the membrane of the cell begins to deform. What causes that deformation of the membrane? Actin filaments. So the actin is causing that action along the membrane. The membrane deforms even more and then begins to wrap around the food particle. Eventually, the two arms that have sort of wrapped around connect back together, and you have a particle of food inside of a vacuole. That's taking particles in. If you were specifically digesting food, this process is known as phagocytosis. Now, if you need to digest that, you've got a vacuole with food in it. What's got to bind up to that vacuole in order to start digesting it and recycling it? What organelle is going to need to do that? The lysosome. So the lysosome comes along, binds to that vacuole, releases its acid hydrolases into the vacuole, increasing the rate of hydrolysis, splitting these individual molecules into teeny tiny pieces. So endocytosis pulls it in, exocytosis releases it.